its own advantages, technology has its own. I welcome you to today's program. As you all know, this is about the Nobel disadvantages. So with this, I welcome you to the first lecture in the series. Today's program. As you all know, this is about the Nobel Prize in Physiology or Medicine, two of the series. And before we start, I would request to say a few words. And before that, let me express my heartfelt gratitude to him. Professor Naveen Gupta, chairperson of this department, to say a few words. Felt gratitude to him because many other hosts have refused to host us. Professor Gupta was the first one who wholeheartedly agreed to host us.
I also apologize for this.
Professor Gupta.
I think I have also hosted 75% of it. 20% we have also not.
I also hosted 75% of it only. 20%. Anyway, the guest of today's lecture, Professor Jayaroop Singh ji, our previous Vice Chancellor, Dr. Arun Grover ji, Dr. Dharamveer, Dr. Mrs. Kia Dharamveer, Dr. Dua, Dr. Pushpinder Kaur, and the faculty members from my sister departments. So I welcome you all in today's lecture, which is a very relevant and prestigious lecture that Nobel Prize in Physiology and Medicine, which has just been given, that the work the scientists who are doing around us related to that, that's what we will be discussing, and they will also talk about the importance of that work, what is the why this Nobel Prize has been given to that, and all those details. And I'm also very thankful to all the members of my other organizers like Society for Promotion of Science and Technology in India, Chandigarh chapter of National Academy of Physicians, with help of whom this lecture has been given. And I'm very happy. Special my comments goes for SPSTI, who are doing. I have attended previous lectures also. They are doing a wonderful job under the mentorship of people like Dr. Grover, and under the hard work of by Dr. Dharamveer and Dr. Kia Dharamveer. So I appreciate all your efforts, and I'm very thankful that it is being conducted in my department, and it also gave us the opportunity to call all the dignitaries to our department as guests. So with this, I once again welcome you, and I request Dr. Dharamveer to start.
Thank you, Dr. Naveen Gupta. I now request Professor Arun Grover to give a brief introduction to this lecture.
So the idea of hosting lectures on expository lectures on Nobel Prizes in all disciplines was commenced during the pandemic in the year 2020. So in the very first year, all the lectures were in online. Second year also it was the same. But from the second year onwards, we added to the Nobel Prize lectures, 6 of them: physics, chemistry, physiology or medicine, economics, peace, and literature. These 6, we added to it a lecture on Abel Prize and also a lecture on Turing Prize. Abel is for the mathematics, Turing is for the computer sciences, and then at the initiative of Professor Dillo, we also added the World Food Prize to it. So in 21, we had actually 9 lectures in online mode. 22, we could hold 8 of them. We missed 1. And last year, Dharamveer ji, president of SPSTI, he said, no, we should go back to the physical mode because you get to meet young people, and so let's do it in a hybrid mode. So we tried to do this series in the hybrid mode last year, and actually we skipped more lectures. We held only 7 out of 9. So this time we are determined that we will do all the 9 in the hybrid mode. So today's is the first one, and the next one will be in the literature, and Professor Pushpinder Syal, who has given the previous 4 expository lectures on Nobel Prize in Literature, will give this year's lecture as well, and that is scheduled on November 20th, and that is in partnership with Department of English and Cultural Studies, and all the other languages department will also join in that. So that's in the Botraj Auditorium of the Punjab University on November 20th. Then we have a chemistry lecture, since that is an area in which we have a lot of expertise all across the institutions in Chandigarh region, but we are going to host it in CSIR Institute of Microbial Technology, because that has a GN Ramachandran Center for Protein Engineering. There is a center which is devoted to protein engineering, and Dr. Beena Krishnan and Sri Krishna Subramaniam, the 2 of them, will give this talk, one on the science part of it and the other the applications part of it. So we are going to have that lecture on 25th of November, where Purnananda Gupta Sharma would be our guest of honor. Then we have an offer of a lecture in physics from Professor Vipin Srivastava from the University of Hyderabad. He is going to travel all the way to Chandigarh and give a lecture in the physics department of Punjab. So this leaves us with 2, and one on economics will be given by the ace economist of India, Professor Arun Kumar of GNDU, who has been our advisor, who supplied us 4 speakers for the previous Nobel Prizes in Economics lectures. And this time he is going to give the lecture himself, and he will travel to IISER Mohali, and that lecture will be done in the evening, either 4 or 5, so that it doesn't clash with the classroom hours. And then finally, the 6th lecture on Nobel Prize in Peace would be given by Professor R.B. Grover, who is a member of the Atomic Energy Commission and who was also the negotiator on Indo-US nuclear deal on behalf of India. So he is going to give a lecture how this strategic nuclear balance or non-proliferation, which has held it so far. We haven't had more nuclear explosions to end or start wars since the nuclear explosions in 45. So he is very familiar with this subject. He is also familiar with the Pugwash movement. So he will give a lecture covering this broad arena, also at IISER Mohali as an evening talk. So this is what the plan for the 6 lectures is, and we are hoping to have all these 6 lectures done by December 10, the day these Nobel Prizes are actually handed over in Sweden. So in between, some more lectures are continuing, in the sense that Dr. Sahu from PGI got the Vigyan Shri Award. So he is going to give an expository lecture at the Dental College of Punjab University on November 11th. Okay, that is the Maulana Azad birthday, the National Education Day. And our guest of speaker on that day is Professor Mukut Mins, the Padma Shri recipient from our region several years ago. So that lecture is also in the PU campus. So lots of lectures. I hope the young people and the faculty of the university will take advantage of this festival of annual lectures in the year 24-25. Thank you very much.
Thank you, Professor Grover. I now request Dr. Nishma, Punjab University, and Nishma is also a fellow of INYAS, Indian National Young Academy of Science. Nishma will introduce today's guest of honor. Thank you, Dr. Nishma.
Thank you, ma'am. Good afternoon, everyone. Myself, Dr. Nishima. I am a faculty at Department of Applied Sciences, UIT, and also a member of Indian National Young Academy of Sciences, INYAS. It's my pleasant duty to introduce the guest of honor for today, Professor Jairoop Singh Ji. He is the founder, Vice Chancellor of Central University of Punjab. Prior to this, he was the Vice Chancellor of GNDU Amritsar. He is also an honorary professor at Punjab University and a member of Chandigarh UT Committee on Higher Education. He was also a member of Punjab Chief Minister's Advisory Committee on Higher Education. Professor Jairoop Singh has been the president of Indian Society of Human Genetics, chairperson of Indian Genetics Alliance, and he is a fellow of International Medical Sciences Academy, fellow of NAMS, National Academy of Medical Sciences, fellow of Punjab Academy of Sciences, and is a recipient of many awards, which include Mental Medal Award by Czech Society and Dr. V.R. Khanolkar Oration awarded by NAMS. He has been honored by Punjab government in 2019 for his contributions to education and human genetics. In fact, he is amongst the pioneers of human genetics in India. He has many scientific books and manuals to his credit. He has published more than 100 research articles and is also a visiting professor to various European and South American universities. With these few words, I would like to welcome Professor Jairoop Singh Ji to kindly come and address the audience. Sir, please.
Thank you very much for your nice words. Professor Arun Grover, Professor Naveen Gupta, Professor Kia Tanvir, and the distinguished professors, retired professors from Punjab University, and Shri Taramveer ji, and teachers and students. I'm really grateful to the organizers for having given me this opportunity to be here today to listen to good, very nice speakers. I have Professor Rajat Sandhir and Professor Sadhan on a very important topic, which has been fascinating, I think, everyone, not only the people of biological, with the background of biological sciences, but also of all sciences. Now, these prizes, Nobel Prizes, which have been given to Victor Ambros and Gary Rupon, they are for a problem which, or a process, I would say, which they found, and that process has been existing for millions of years. It's not nothing which came up new. It existed, but it was only in 1993 that they could decipher it, they could find out and say what is the importance of this one. If you look at nature, nature is very conservative, extremely conservative. If you look into the inheritance of all the organisms, including human beings, everyone, what is basically involved are just 5 elements. We have carbon, hydrogen, oxygen, nitrogen, and phosphorus. That's the end of it. They are all the combinations and permutations and combinations of these that you make all types of DNA and all types of RNA and everything else. And if you look into the composition of the human body, just add one more element, calcium, and 99% of the human body is taken care of. Now, when we look at the start of the life, at the time of fertilization, you all know, it starts from one cell. And that time of birth is 1,025,000,000,000. And an average adult has almost 30 trillion cells. And all these are supposed to have the same genetic constitution, same DNA with which they started with the first cell. So what happened from the first cell? We have all the organs developing, all the different types of cells developing, and the consensus is there are more than 200 types of cells in the human body. And this figure, some they indicate to be more than 6,000, because last year in the brain itself, they found more than 3,000 types of different types of cells to exist in the brain. And when we look into the number of genes, there was an estimate in 1964, there are 67 lakh cells, 67 lakh genes in the human body in each cell. And now in 24, the figure is now reduced to 19,411. Now these 19,000 odd genes, they are producing more than 5 lakh of proteins. And those proteins are ones which are all making our organs and everything else. And number of organs, we are not sure about how many organs a human being has. Till last year, it was 79. And then they found another salivary gland, new one. So now they claim it to be 80. And every year, at least there are 1 or 2 reports which claim finding of new organ in the human beings. And the problem comes in that all this 5 lakh of proteins, how we are achieving with just 19,000 genes. So there has to be a process in which all the genes are not active in each organ, and specific genes at specific times, they are triggered to produce a type of protein which is needed. So in the 80s, came the transcription factors. The discovery of those transcription factors, they said the problem is solved now. We know how things are being regulated. Till 1993, the discovery of Victor Ambrose and his team. And what they have discovered, we'll be listening in detail from the 2 learned speakers, but I'll just tell you about the organism, C. elegans. I hope this system works, and one of them shows the movement of this 1 millimeter long soil nematode, which is transparent one. As they say, is a miracle nematode, miracle animal. And it has a very elegant, that's how the name comes. It's from the Latin word, elegans. C. elegans is from the Latin, means elegant. Its movement is very elegant, very sinusoidal one. Now, this small C. elegans, small 1 millimeter long nematode, this has just very interesting. There are 2 sexes, female and male, but all the females, they are in fact hermaphrodites. They don't need the males. They produce their own sperms, fertilize them with their eggs, and system goes on. But 1 in 2,000 or 1 in 1,000, there is the frequency of male appearing also. And both they have different type of chromosome combinations. The hermaphrodites, they have 6 pairs of chromosomes. The males, they have 5 pairs and 1 X chromosome. constitution. And their genome, it contains more genes than the human beings. And it's just, if you look at the number of cells, 959 somatic cells in case of hermaphrodites and 1033 in case of the males. And each cell has been studied in detail. Now this 1 millimeter miracle, now that's good. The system is working. Now this is the micro system working, the micro things. The micro RNA is functional now. This is how it's being regulated at the micro level, all the genes. What they found was that this small pieces of nucleotides, which are 21 to 25 nucleotide long, they actually control the functioning of the RNAs and then subsequently the production of the proteins. And they actually regulate all the systems, all the organs, all the cells in all organisms. Earlier, it was thought that this is only confined to the worms, but it was later on, it was found that it is a conserved one. These microRNA, they are highly conserved and they exist in almost all organisms. That's why these two people got their Nobel Prize on their finding of this one. And actual finding was in 1993, on the basis of which they have got the Nobel Prize this year, on 7th October. And I also am quite eager, like all of you, to listen to the two learned speakers. And I once again thank the organizers for having given me this opportunity to be here with you. Thank you very much.
Thank you, Dr. Jeru. People like me always wonder why C. elegans and why not D. elegans or any other organism for that matter. So you have thrown some light on that. And I'm sure the speakers will carry it forward. And now I request Dr. Ravneet Kaur to come and introduce the first speaker of the day, Professor Rajat Sandhir. Ravneet Kaur herself is an avid researcher from Zoology Department. Ravneet.
Thank you, ma'am. Our very own and dynamic professor, Dr. Rajat Sandhir. He has received his MSc and PhD degree in biochemistry from PGI, Chandigarh. He has been in the Department of Biochemistry, Punjab University for more than 20 years. He has 30 years of teaching and research experience. His research interests are to understand the biochemical and molecular mechanisms involved in development of neurodegenerative conditions like metabolic encephalopathies, dementia, and brain injury, with a particular interest in investigate the role of oxidative stress, mitochondrial dysfunction, and alteration in permeability of brain blood barrier. In addition, his interest is also to identify neuroprotective strategies that could ameliorate neurodegenerative conditions. He has more than 200 papers in his credit and has mentored 3 dozen students for PhD. He has an H index of 52 and I-10 index of 154. He was been awarded with K.T. Shetty Memorial Orientation Award in 2021 by Indian Academy of Neurosciences and Miss Abadi Mahal Award 2022 by Indian Academy of Biochemical Sciences for outstanding contribution in the field of neurosciences. He is also Fellow of Indian Academy of Neurosciences and Indian Association of Biomedical Scientists.
He is also recipient of INSA Best Teacher Award 2022. He has been enlisted among World 2% Scientists for the year 2020, 23 and 24, released by Stanford University. He is on editorial board of many international journals. He is Dean of Indian Academy of Neuroscience and Vice President of Society of Neurochemistry in India. Presently, he is the Principal Investigator of North Region S&T Cluster sanctioned by PSA Government of India. This is just a brief introduction, and I would request and invite Rajat sir, please.
Very good afternoon to you all, and it's a pleasure coming back to the forum of SPSTI. Unique initiative and good cause that they are spreading the word of what the Nobel laureates do. And as I would say, this is a forum where I've come a couple of times, always enjoyed, learned along the process. And when I say I'll be discussing or sharing the work of Nobel laureates, so you can understand sharing the work of Nobel laureate is not easy, but I will make an effort or endeavor in that direction. And so I also did some work on microRNA. That's why I volunteered to talk about it. Although Dr. Sadhan is here, who is actively pursuing that interest. And since there are two speakers, you will also see some kind of overlap between what we discuss. Because the primary purpose is to talk about the work of the Nobel laureates. And as a new person from neuroscience, I believe in reinforcement. Reinforcement always helps to learn things even. So if you get a double dose of anything, that means it's reinforcement of things. And so if I fumble, I am standing in front of my mentors, Professor Grover, Professor Dharamveer, and Dr. Jayarup, whom I consider them myself as my mentors, and they've nurtured me for last many years, I would say. And so I've titled my talk as microRNAs, tiny regulators with great potential. So we'll talk about the journey of microRNAs from discovery to some of its applications. So talking about the work of Nobel laureates, which started in 1970s, this work in this direction started in 1970. So if you have to tell it in 30 minutes, it will be a challenge also. So I'll just give you a flavor, an overview of what is the work all about. Can I have the first slide? So this is the picture picked up from the internet. You all seen this picture. Victor Ambrose, Gary Rukan, and the work that got Nobel Prize about discovery of microRNA and this role in post-transcriptional gene regulation. So they say one of the best example to see gene regulation is to go to a butcher shop. So I'm quoting the words from David Liachman, who said, if you have to see differentiation of tissues, the best example of laboratory is to go to a butcher shop where you see liver, you see different organs, how they differentiate. Dr. Jayarup also told there are certain, everybody carries the same kind of DNA, but how the DNA changes into different cells, different organs is all dependent on gene regulation in each of these cells. Next slide, please. So we all familiar with this flow of genetic information. We've been reading in our schools, and we said DNA moved from RNA to protein, and then protein is what gives rise to cell types. And Dr. Jayarup told 20,000 genes, and he even brought down this number to something 18,000 plus. And so when we were students, I think we were hearing the numbers were higher. So now we're coming down in terms of numbers of these genes. So this is the simplest way of flow of information that we can see in any organism. DNA, messenger RNA to protein. And this whole dogma, which we call a central dogma, is kind of redefined with the discovery of these two Nobel laureates. Next slide, please. So let's try to know about these two Nobel laureates. Victor Ambrose, born in 1st December 1953, Hanover, New Hampshire, and his present affiliation has been at University of Massachusetts Medical School, and did BS in biology in 1975, PhD in 1979. Doctoral supervisor was David Baltimore, who is a Nobel laureate. I just wanted to emphasize on this part. Look at something more interesting. Postdoctoral training was in the lab of a future Nobel laureate. Then he did not get this Nobel Prize at that time, was H. Robert Horwitz, who introduced him to C. elegans in that time. And I will be talking about C. elegans also. Next is the other Nobel laureate. The half of the prizes shared by Gary Ruvkun, born in 1952, Berkeley, California. Affiliation is Massachusetts General Hospital, Boston, and Harvard Medical School also. BA in biophysics, PhD in biophysics. Did doctoral studies in bacterial nitrogen fixation genes. Something that connects both of them is Robert Horwitz. Both of them did postdoctoral work with Robert Horwitz, and they were trying to understand one of the genes, that is Lin14, which is part of their discovery of the Nobel Prize. Next slide, please. And this is their mentor, Robert Horwitz, who got Nobel Prize in 2002. So it's two postdoctoral training, both get Nobel Prize. And he got this Nobel Prize for discovery of genes that regulate programmed cell death. We all know when the tadpole, the best example of programmed cell death is the disappearance of tail from tadpoles. And there are other examples. I just took a classical example because we've all seen that. This all happens in biology. We lose cells in a programmed manner, so that this is a normal process of development. During brain development also, we're born with more neurons, we lose neurons. This is programmed cell death. Can I have the previous slide? So he only introduced these guys on working with C. elegans, and I'll be talking about what is so important about C. elegans, why it is a good model organism in my next few slides. So this is C. elegans, transparent looking worm. You can see each and every part of this neuron. You can see muscle, you can see gut, you can see other parts. Interestingly, I work in the neuroscience. It has 308 neurons, and out of these, 8 are dopaminergic neurons. And you can see these 8 neurons very, very clearly. So if you want to look at something that is related with, let's say I want to create a model of Parkinson's disease, I want to see if these cells or Parkinson's dopaminergic neurons are dying, I can visually see that with a bit of staining techniques. So this is the strength of this model that they used. And this has been an excellent model system for many, many reasons, that it shows more homology with any model system that you could see with humans. And you can create disease models in this also. Next slide, please. This is the work that Horwitz did, where he discovered this programmed cell death. Programmed cell death, what happens is normally the cells will be live. The first top picture indicates live cells. And what you see is there are 3 components from C. elegans, SADNI9, CD9, which is the apoptosis suppressor, CD4, adapter protein, and then you have inactive executor molecule called caspase. So once the cell decides it has to go under cell death, what happens is one protein comes into play called EGL1. EGL1 was discovered by him, which once it binds, it then makes the execution signals available, and the cell undergoes apoptosis. Next slide. So what these guys did, and Ambrose and Rukun did, was Ambrose cloned the Lin4 gene, and he found this Lin4 gene does not make any protein, and it codes for 22 nucleotide long sequence, which we now call it as microRNA. What did Rukun do? He cloned Lin14 gene and found that Lin4 regulates Lin14. Lin14 is a transcription factor. That means and these are genes that control different stages of development of C. elegans. And so this was the first proof. And what else they found was this Lin4 binds to untranslated regions of Lin14. That means it's binding somewhere not in the coding region, but non-coding region of the messenger RNA also. And there are multiple sites where it can bind. Next slide. This is the heterochronic worm mutants. Heterochronic for the reason you can look at genes that play a role in development of C. elegans. This is the top one is wild type C. elegans, normal morphology. Lin4 mutant. Some of the morphological changes happen in this worm. Lin14, you can see the worm is shortened, and these don't function as well. I'm not getting into the morphology of these. Maybe the next speaker will be talking about that in more detail. Next one, please. So what they did was they said Lin4 controls Lin14, and then both of them, their discoveries were connecting. Lin4 was discovered by Ambrose, and Rukun showed that Lin4 controls Lin14, and which is developmentally regulated genes. And what they showed was, you can see the bottom panel where you see Lin4 binding to Lin14 messenger RNA. The binding is not perfect. Some of the region forms loop. So this is something that I would like to emphasize here. And both published their research work in same year, 1993. Dr. Dept also mentioned that. Both came back-to-back papers in Cell in that year. Next one, please. Rukun then discovered another small RNA called LET7. After 7 years, another miRNA was discovered called LET7, and this RNA was small RNA was evolutionary conserved in all species. To talk about humans, to talk about amphibians, reptiles, everywhere you see them in most of the organisms. And whereas Lin4 was not there in all organisms, so you discover an RNA, microRNA species, which is evolutionary conserved, and for 500 million years this RNA species is conserved. And now we know there are equivalents of LET7 RNA in many organisms with different names. In humans, we call these as MIRs, and the nomenclature varies because the C. elegans RNAs are called LINs and LETs, and we go to a different nomenclature there. So, based on the discovery that they found that when they found a second species, then they thought it might have bigger roles to play than they thought in the first experiment, where they just showed one gene being regulated by them. This is temporal regulation of microRNAs, Lin4 and LET7, the two that were discovered. So once you see Lin4 goes up, what you see is Lin14 RNA is gone down. Vice versa, when you see LET7 gone up, you see Lin14 is gone down. So these are all changing during developmental stages of the C. elegans, indicating that these are genes that regulate development of these organisms. Next one. So what is microRNA, just to make the rhythm? See, when we come to a room, we know there is an on and off switch. So for all genes, we need this on and off switch. So on and off switch means I need the fan at full throttle, full speed, I will have this on and off switch. But if I have to have a final control, I need something like a rheostat. Physics people are there in the audience, they will understand rheostats. And also when we were students, we did rheostat experiments where you have resistance applied to the circuit. So what is this microRNA is doing is to fine-tuning the amount of gene that has to be transcribed. So if I have to have a fan at 1, if I have to have a fan at 3, you need microRNA species. Next one, please. So we say we call all these microRNAs, and there are other species also, as non-coding RNA because they don't make any protein. Earlier, we used to think these are all junk. We don't know till we do not know the function of them. So there are two kinds of these non-coding RNAs based on their size. Some are called small non-coding, some are called as long non-coding. The size is that decides whether we call them as small or whether we call them as long. Length more than 200 nucleotide is called long, and lesser than 200 nucleotide is called short. And microRNA is one kind of these small non-coding RNAs. And I know Dr. Das and IISER Mohali is working on long and non-coding RNAs, and probably I was asking him maybe briefly tell about his work that he does there. And within the small non-coding RNAs and the small ones, we have some that play structural roles. For example, they're component of ribosome protein synthesis machinery. They are acting as adapters for amino acids. tRNAs, and there are a few other functions that they carry out. So our emphasis mainly today will be to talk about miRNA, and you can see the structure of this miRNA. It's indicating hairpin kind of structure at the very bottom of this slide. So within the non-coding RNA, this left side tells you microRNAs can be gene silencers. They can silence certain genes, and the dose would be important, that we say. And then the long non-coding RNAs also. Nowadays, we are knowing there are another species that has come up called circular DNA RNAs also, which the role is emerging. Probably we'll hear another Nobel Prize given on some other species of these non-coding RNAs. And next one. So non-coding RNAs are adding to the complexities of the genome or eukaryotic genomes also. We have them. So I will not use this figure, but I'll just have the heading here because the numbers are evolving. And when I was doing research on microRNA, probably I knew there are 800 species of microRNAs known in humans, while we know there are more than 2,000 species of microRNA that are known. And they primarily, we know they are controlling protein coding RNAs only. And for example, C. elegans, it said there are 73 species of non-coding RNAs. And in humans, at the time this table was there, probably this number had only few, but now this number has gone considerably higher. Look at Saccharomyces cerevisiae, 29 of these non-coding RNAs, and we know they play important role in many of these functions in the cell. Okay, this is a recent data from MIR database, where they said there are 38,000 hairpin precursors, 48,860 microRNAs, and across 271 organisms. This is what the database has, MIR base. And even the viruses have microRNA species. Next one, please. So what are the characteristics of microRNA? Small, non-coding, we said that. Size is 19 to 22 nucleotide long. Repress activity of complementary messenger RNAs. So need to have binding sequences for repression of the activity. Regulate 30% of the mammalian gene products. And we can expect one microRNA to control number of genes. We can also expect one gene to be controlled by number of microRNAs also. And you can change the phenotype of an organism by modulating single microRNA species. I'm not getting into those kind of things, but you've seen that in the C. elegans experiments also. The phenotype changed with one change in the gene sequence. And we know it's present in almost all organisms, and many are conserved between vertebrates and invertebrates. We also talk about that. I will also make one point across here is they are much stable compared to other RNA species. Since they are very stable, that adds to one more property. They can be used as biomarkers or diagnostic markers for a disease also. This is the structure of miRNA, hairpin structure. But if you see the top of this picture, it says miRNA. That means precursors of the two stages of this microRNA formation. One is pri and the other is pre. So that means if you truncate this molecule, you will be left with some middle portion of this molecule, which is single-stranded, which will be the mature miRNA species that will be functional species. So that means we have stages in the biosynthesis of this microRNA species. Next one, please. This is a timeline of discovery of miRNA. This slide I had a few years back. So you can see something very particular in this particular slide. 1993. This slide I picked up from some literature which didn't mention about Rukin's discovery. It only talks about Victor Ambrose. So 1993, we know lin gene was discovered. And before that, we all know about the discoveries that were happened. So I will just emphasize on the landmark things that are related with this. miRNAs are dysregulated in cancer. So a lot of interest is there in role of microRNAs in cancer. Diagnostic markers, I said, 2004, 2006. We are saying they regulate mammalian cells by epigenetic mechanisms. And They are also regulating transcription by, and these are discoveries continuing. And this is an old slide that I picked up a few years back. Next one, please. This is about biogenesis. There are different stages of biogenesis, and there are proteins or machinery involved in biogenesis. I will just, so I said, I talked about this precursor of microRNA. I said 70 nucleotide hairpin transcript pre-miRNA, which is cleaved by an enzyme called DROSHA. I want to talk about this enzyme because there is a reason. I will be connecting it later also. So the DROSHA is a crucial component in this microRNA species. Then we also that does that. The other components of these machinery, there are enzymes like IISER, which will be important. The last part says 700 human genes, 490 mouse genes, 100 possible human sequences of microRNAs that have been detected. And in wet lab experiments, you could only test 120. So that means the story about knowing the roles is still emerging. Next. So microRNAs and the roles, look at almost everything is taken care by microRNAs. Cell proliferation, cell differentiation, development, epigenetics, insulin secretion, apoptosis, stem cell, blood cells, cancer, fat metabolism. So that means versatile kind of roles are there for these species. This is again talking about timelines. And if you look at this, after 2005, what you see is called 2002, around 2002, we see microRNAs in cancer. Then we started coming up with approaches which will be trying to modulate the expression of microRNAs so that we can use them for therapeutics. And these have been tried in cardiovascular disease, and a lot of them have been shown to play a role in cardiovascular disease. Next one. All right. So look at one of the components in microRNA biogenesis, IISER. If you remove that from zebrafish or mice, it is embryonic lethal. Had that not been a fundamental function of these, the zebrafish or mice would have survived. And then if you remove individuals of group of microRNAs in fruit flies or mice, you create strong phenotypes. That means fundamentally they're doing something which is crucial for their survival and function. Look at something more interesting. Previous slide. Okay. There is a condition called IISER1 syndrome, which is a rare inherited disorder caused by mutation in this gene, and which results in tumors in kidney, thyroid, ovaries, testicles, and brain and eye and lung. So that means if you remove one of the components in biogenesis, it leads towards cancer. So that means you have a clear evidence of this playing a role in this. Next one. All right. So now I just want to highlight about some specific miRNAs in human disease. Mutation in miRNA 96 are associated with progressive hearing loss. Mutation in miRNA 184 causing a condition called Edict syndrome, a rare eye disease with iris hypoplasia, endothelial dystrophy, and congenital cataracts. Mutation in MIR 145P result in congenital skeletal disorder. I've just picked few examples to illustrate that they are important in biology and in have a role in disease conditions. Advances nowadays, we know that we are using them for developing diagnostics, therapeutics for disease, and for conditions like metabolic disorders, cardiovascular disease, neurodegenerative disease, and cancer. Next. This is some flavor of the kind of work I did many years back, just to illustrate. So this is miRNA traumatic brain injury. On the left is the control brain of a mouse. Right is the middle lane is the brain of a mouse which has been injured with a trauma and 3-day post-injury. What you can see is the species of microRNA change in these two brains, and you can see which one of these specifically change by carrying out ratio analysis of these. Next slide, please. So I want to show some of the mRNAs we saw, and this work was almost done in 2008 or maybe 6, 7 or 8. So we could see many of these mRNAs were changing in these brains. So I just want to highlight on one microRNA, maybe number 4, and MMU indicates mouse microRNA mid 21, and it was changed 2.7 fold. We picked up this for the reason a lot of people had looked at this microRNA and they called it as oncomere because it plays a role in cancer. So we also found this RNA species were going up in injured brain. Next one, please. And we published this paper long back where we showed differential response of MIR21 and its targets. So we also looked at the targets of this miRNA, which will be playing a role in apoptosis and in, I would say, in vascularization and a few other things in aging brains. And we found that there is a differential response of this RNA in the aging brain versus that of adult brain. And now we say miRNA is one that changes in a lot of conditions. I said cancer, I saw it in brain injury. So I just found an article which says biomarker across spectrum of disease. So we can see one of the RNAs just change in many of the disease conditions. And I said this is called as oncomere. Next one. Interplay, another article from us which said interplay between metabolism and oncogene process, role of microRNA. So we said it does play a role in cancer, and a lot of strategy being tried on this. Next one, please. So talking about its applications, we said diagnosis, primary screening, diagnostic marker. It can differentiate between early stage and late stage patients of cancer and other disease. Prognosis can be good prognostic marker. Nowadays, focus is on developing therapeutics. You can have antisense miRNA drugs, which can interfere with the action of these, or you can have mimics of microRNAs, or you can overexpress them, and you can have other techniques to modulate the expression of microRNAs. And these are all, some of them are at preclinical stage, some of them are at clinical trials also. Next one. So we also had some analysis done. This was again we used some in silico predictions in 2000 prevention and treatment of neurodegenerative disease. What we picked up was we did data analysis, picked up the RNAs that change and that are unique signatures to Parkinson's disease, Alzheimer's disease, and few other conditions, so that we can use them as measures to target them and maybe even use them as diagnostic markers. Next one. This is a very recent work where we are trying to show you can use microRNA 137 in Alzheimer's disease, and these are studies that are still ongoing with PGI, where we want to maybe validate that if that can be used as a tool to predict early diagnosis of Alzheimer's disease. And next one. So in a nutshell, I will say new dimension in gene regulations. MicroRNA is playing a critical role, and they're critical for animal development, adult cell types, differentiation, and in multicellular life, these are very, very important molecules. And we are understanding they play an important role in pathophysiology, and which still is emerging. And we also say they will be potential targets for therapeutics. And Lecture-1 family has been shown to be playing a key role in suppression of tumor growth. And next slide. Since there is an audience, I would like to say that I represent this Northern Region Science and Technology cluster. Please do visit the website of this cluster. And next one, please. So there are 5 thematic. This is the only regional cluster, and there are 5 thematic areas which are indicated in the next slide. And there are different programs where each one of us can take benefits. Students can write grants, funding is available, and thematic areas are agriculture, food processing, waste management, waste from wealth, sustainable mobility, green energy, indigenous technology development and optimization, pharma, healthcare, and medical devices. Thank you all for your attention. And any questions, I can take that. Maybe or we can have it at the end.
Thank you. Thank you, Professor Rajat Sandhir. That was rather overwhelming, wasn't it? But that's the subject of today's talk. And now I again request Dr. Ravneet Kaur to introduce today's guest speaker.
So our second speaker is Dr. Sadhan Chandra Das, has received his MSc Zoology from University of North Bengal and PhD degree in École Norme Supérieure de Lyon from France. Initially, he joined postdoctoral fellow at Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore, but soon moved to Beckman Research Institute, California, to complete his postdoc. Presently, he is assistant professor and involved in teaching and research at Department of Biological Sciences, IISER, Mohali. His research interests are to understand signaling, transcriptional and epigenetic mechanism in diabetes, macro and microvascular complications. He has investigated and published work related to molecular mechanisms of epigenetic factors and long non-coding RNAs that promote inflammation and cardiovascular disease. He works on the hypothesis that the distinct cell type can differentiate into different cell states due to diabetes, cardiovascular diseases, or high risk factors that can be addressed by understanding epigenetic mechanism. His long-term goal is to understand how diabetic conditions mediate altered functions in various cell types and to identify possible therapeutic strategies to mitigate impaired wound healing and cardiovascular diseases in diabetes. It is he is utilizing next generation sequencing technologies, advanced molecular biology, multiomics approach, including cell cycle transcriptomics and epigenomic analysis. His research is funded by DBT, Welcome Indian Alliance, and Serve India. He has received many grants, honors, and awards for his research and published research articles and reviews in highly reputed journals and conference proceedings. He has also written book chapters and delivered number of talks at various platforms. He is on the editorial board of ATBB 2023 to 25 under American Heart Association and is member of many international societies. This is the brief introduction, and I wish to invite Dr. Das to please deliver his talk. We eagerly wait for it. Thank you.
So very good afternoon, everyone. I think thank you for a very kind introduction. I don't think so. I haven't reached that far, but I think I should record that and tell everyone what you talk about me. Okay. So first of all, I would like to thank the organizer for giving me the opportunity to talk, especially in this platform, to talk about especially the Nobel Prizes that is rewarded in the year 2024 for the discovery of the microRNA and its role of post-transcriptional gene regulation. And off note, I like to thank Professor Kia for inviting me and just deliver a talk. And before that, I like to thank before Professor Singh and Professor Sandhir just to make it so easier for me, so I can go into much more detail and make a little bit maybe complicated, but I'll not bore you. Okay, so let's learn more about the microRNA and everything. Is the online is on or online is off?
Online is on.
Online is on, right? So because some of my friends they wanted to hear it. So okay. So and today's talk is basically focused on the non-coding portion of our genome, that is the microRNA. So can I go back to the next slide, please? So here, do I need to stand here or I can go there?
No.
It's here, right? Okay. You can hear? That will be easier. So earlier, Professor Sandhir says our genome transcription, right? So in our genome, basically, genome is composed of various forms of DNA and of RNA, and also different regulatory elements. What those regulatory elements? Regulatory elements can be anything, the DNA or the RNA or different post-translational entities. So if you see in this slide very carefully, this is the two end of it. On the extreme left, we say it's a very simple organism, and here is a multicellular organism. So look at very carefully in the simplest organism, they are only composed of single cell. But when we move forward, the evolution, they form a multiple layer. That means they are a very complex organism, right? So what you see is even while they are unicellular organism, they have many coding genes and this divide of the non-coding genes. In the evolution period, if you move more in the higher organism, what you will see is the reversal. There will be less in the coding gene and more in the non-coding gene. How this happen? Maybe there is a selection procedure happening that can select maybe only a few coding genes, but at that time nobody knows what is the role of all the non-coding portion of the genome. That's what we are saying here. So that can be next and back to the next. So now I'm going to ask what is this coding DNA? Coding DNA is mainly code for protein, or it carries the genetic information. Whatever the things we are talking and everything is because of our DNA, and DNA is ingrained in our chromosome. All of people are spaced here, we all have the same kind of chromosome, and we are carrying the same genetic information, but we behave differently. Why? I'll explain more. And rest of the other genomes is composed of the non-coding DNA as considered as junk DNA. Junk or very kind of slang word earlier. But later, this junk DNA can consider as treasure island because that's how that discovery has been made. And people are thinking, what does it do? Not genotype. And another important thing you have to know, what is the C-health paradox here? So C-health paradox is this. We have a different organism, but that doesn't correlate with complexity. I will explain. Let's say in a human you have our genome size is big, but we also have the same 20,000 genes. That's one of the examples from the C. elegans as a linear demarker. The picture is saying that. What is saying that is a very similar number of protein coding genes to that of our human or the mouse. That is approximately 20,000 genes. Even though their genome size is varying, the size by 34. That means C. elegans has a smaller genome. But our genome has 34, but they have a similar number of genes. It's suggesting when the organisms are very small entity, they have more coding genes, less non-coding. When you move toward evolution history, that is the increase in the non-coding genes. Now we'll be learning what is non-coding portion of the genome history of the next piece. So now next. The biology has
mRNA or the messenger RNA, correct? Then this messenger RNA go into the cytosol or the cytoplasm, which get translated into the proteins. And the proteins carry out our cellular and the molecular functions. Next, please. Now, I think the same slide what Professor Sandhir said, but I'll explain it differently. So here, I want to talk about the regulatory mechanism and how the each of the regulatory mechanism is complex, and there is a different control mechanism. You can control everything. How? Because I say our human genome has 20,000 genes. So technically, we have a number of cells. It should have a more or less similar cell. But why your neuronal cells or muscle cells behave differently? So that suggests your neuronal cells and everything have a similar sense of gene, but they are not functionally active. When it's required, only it will exert its function, right? That means this procedure is very tightly regulated at the various different steps. One is maybe transcription, one is translation, on that you made the post-transcriptional step. So the next piece. So how the gene regulation will happen? The
This is gene regulation is tightly connected phenomena which is controlled by different regulatory mechanisms. What are those regulatory mechanisms? You can have a regulation the transcription step and the translation step. You know the protein is always not functional. It has to go to the post-translational mechanism, then only be functional. But let's say if you have a DNA or the RNA that have a transcription, right? DNA making to RNA. Sometimes the RNA is also post-transcriptionally modified, right?
Is the Nobel talk today, that your RNA modified by the post-transcriptional mechanism and how it controls the thousands of gene expression in an organism. Next, please. So now, this is the discovery of the different genetic regulatory mechanism throughout the year. Can hit one more. So one more. So here in the year 1951, the first of the discoveries made by the Barbara McClintock. You know, right? The maze. It's called the jumping gene. Right? In the genome, your genes are jumped from one place to another place and control your different functions, right? And they play a very, very important role, and they are actually situated in the non-coding region. At that time, people do not know what are they doing. But later, in 1961, Jacob and Monat discovered the lack of peron. It's very important discovery. Then you move forward, then Professor NG said there's the epigenetics mechanism. How the epigenetics came? Because they are the discovery of the histones, okay? Then there's a different epigenetics mechanism, SCAM, DNA methylation, and so on. I'm not going to discuss that. But major two important discoveries happen on this rectangular. One is the 1984, the discovered enhancer by the Banerjee et al. Okay, what is the enhancer? Enhancers are DNA that is situated in our genome that control the gene expression in a larger extent that is situated from far away from the promoter of a particular genes. And that time, before that in 1990, they also developed another form of RNA that is called long non-coding RNA that Professor Sandhir nicely explained. And I will not talk about it. By my laboratory majorly work on this. And the later, 1993, they discovered the microRNA by Victor Ambrose and the Röpken. That led to the Nobel Prize, okay, after this year. But it began long time back. But here, what you are saying, they are all belongs to the non-coding region, right? And exact multiple functions. The next please. So now here I'm to say why the number of microRNA, it may be the decreasing if you go in the higher evolutionary rates. That can be explained by here. Why? This is the human genome. Let's say I said there's approximately 20,000 genes, 19 something, and rest of the areas are composed of the non-coding portion of the RNA. There are different class of RNA, antisense, link RNA, and so on, so on. This is not the topic of the discussion. Next, please. So today's talk, I'll be talking about the microRNA gene. This microRNA are 18 to 25 nucleotides long and regulate the post-transcriptional gene expression, right? If you see in this figure, this is a human, right? This is a human genome project is come much later. And the 98% portion are the non-coding of the gene. And one of the things we'll talk about the microRNA. This is basically 22 nucleotide long, and they exert different function, or they are involved in the different developmental process and so on, and or the different disease progression. So if you wanted to learn more about the junk DNA and everything, you can simply read a book by the NASA Carey. It's written in a very layman terminology. It's a wonderful book. She is a fantastic writer. She wrote books in the epigenetics and the non-coding RNA. And there, people have shown this is not anymore junk DNA. It's called the treasure island because all the important discovery is coming from there. Okay, next please. So now, here, I'll give you a brief discussion about the lower organism, the higher organism, and how the evolution play the role. So if you treat the lower organism, especially if you know zoology, classification, anyone, then you say they are the protozoa, ctenophorans, right? These are the three things. There's no microRNA gene. And the other important thing, there's no microRNA pathways. How they control, you do not know. If you move in your clade or the cladogram, you go to the higher organism, then you will see the fundamental things is changing. First thing, if you go at the bilateral, we are all at the bilateral element, right? Because you have a symmetry. What you see, the expansion of microRNA repertoire in the bilateral regions that associate with emergence of triploblastic body plan that we have, and that gains the specialized cells and tissue type because of the microRNA. I'm not reading their phenotype here. Then you move little bit much higher organism, then what you see, that microRNA gains during early vertebrate evolution and are associated with expansion of immunological capabilities and acquisition of adaptive immunity. But let's say you want to move in the primate and human. What do you see there? There is a primate-specific microRNA were recruited for neurodevelopmental roles during human brain evolution. That may be one of the important phenomena that maybe the Nobel Committee thought is ought to give a Nobel of the microRNA discovery. That I'll come much later. Okay, next please. Now, in the researcher people, you do research, right? We cannot use on human directly. You have to use different model system to the basic research. And always you have to remember the basic research is still a fundamental thing. You have to fund basic research. Otherwise, this discovery will not be made. So what are the organisms? There are thousands of scientists in the world. They are not working on one model system, right? They are working on multiple model system, especially in the biology. Very simplest model system is bacteria, it's a single cell, but still people are doing a lot of work, right? Then the yeast. Then today we'll talk about the nematode, there's a fruit fly, and the cell culture in the mouse. Next, please, next. So another next. So you can see that well characterized organism with a lot of tool available to us in the modern science. That's why we are able to do a lot much thing. If you see later, I'll explain these two paper of the, at that time only they have how many panel figure, 3 or 4. That means the Nature paper, right, back to back, and also gave you the Nobel Prize. But now this panel, it will not go anywhere. You will be not able to publish some maybe abstract and some scientific meeting, because science has progressed so much. But that model system is, this is not by means of the exhaustive list. There are other model system also, right? And go next. So in today's talk, I'll basically, we'll mainly talk about this the beautiful organism that's called nematode or the C. elegans. This is only very tiny, it's 1 millimeter long. And they have a transparent body. You can see under the microscope. You don't have to do very fancy microscope. We'll see what is inside, right? Right? Even you can see they are hermaphrodite, they are male or not. You can see they are gonad structure. And the most important thing is it's a very less money consuming. Why? Because you will be able to finish your experiment so fast. They only survive maybe only 3 weeks, and they can make another copy at only in the 3 days. And they feed on bacteria. You don't have to even feed. You just give your bacteria and they will work. And whatever the thing you want to do is give a bacteria. You want to silence and express, you give in the bacteria and feed them. They'll eat, and after that you'll see the wonderful phenotype. So this is the beauty of this organism. Next. So using this model system, you know how many Nobel they produced? 4. Can you imagine? First model is came in 2002. What they do? I think Professor already mentioned the Horvitz. It says the genetic regulation of organ development and the programmed cell death. Or they work on the different dead genes, right? They developed a very important on the development. And this is one of the things, Sidney Brenner, Horowitz, and the Solston. Later, the next Nobel came in 2006 for the discovery of siRNA to silence a double-stranded RNA. If this discovery is not done, maybe this microRNA will not get the Nobel, because you will not know the mechanism. It's very important discovery. And you will know these people are connected to each other. They are mentor or the student. I'll come later. Okay, I'll come in between. The Nobel also a lot in the chemistry. How? But they use CA elegans in the model system. For the GFP, you leave the green color, right? This is red, but it's the green fluorescence color. It's also discovered in the C. elegans, right? In the molecular biology and everything, you put a color, you tag something, and look and go in the microscope and see, how is this color? This discovered there. That also rewarded as a Nobel Prize. And later in 2024, this Nobel study is called for the discovery of microRNA and the role in post-transcriptional gene regulation by Victor Ambrose and the Gary Rapkin. Now I'll give a kind of their analogy. So Victor and Gary are the postdoc of Robert Horvitz lab, right? I'm not going his PhD at the Baltimore lab. Baltimore is close to my institute, Caltech. Now this guy, Craig Mello, is the most youngest one among them. You know, Mello was a graduate student when they have joint lab with the Victor Ambrose together. They used to work day and night and see how this thing's working. So all this Nobel laureates, so they are related. Maybe if you want to get a Nobel, maybe you go with the Nobel laureates lab. I do not know, it's a joke, right? So it suggests it's a powerful tool to study all these things. So now on, I'll just talk about this work with the Victor Ambrose and the Gary Rapkin's work. Next, please. So I don't want to explain much. It's already covered. They got a Nobel in 17 October 2024. Go next. So now what I wanted to say, what the previous aspect didn't cover, this is done in the Horvitz group. Why this is important? This is very fundamentally important. That's one of the phenomena called the fixed cell lineages. That's only first time is shown in the C. elegans. What is the fixed cell lineages? Let's say I give an analogy. We live in Chandigarh, right? You want to go to Mumbai. So there's a direct flight. You can book a direct flight, maybe expensive. I do not know, cheaper. Or you can give a multiple break journey to Bangalore, I don't know, to Chennai. So different route. So C. elegans doesn't like that. They always like the direct route. Why? Because if this H1 is a different cell, just forget about the name. This always make the same cell types and same generation after generation, right? They have a different step, larval stage 1, 2, 3. So they are very fixed and direct. Also, when you cross them, what they will make? So they will make a fixed number of genes. Let's say they are making 100 genes, the next generation they will make only the 100 genes. So what will happen? Let's say in the mutation or something, you change one of the things, right? I take your one thing. Maybe your 100 genes is not there or 99. So if you do the genetic screen, you will easily find that, okay, there is a mistake. That's why it's so powerful. That's why they found all this thing, will figure it out. So it's suggesting the Horvitz lab, this lineage generates a fixed number of progeny of the cells, or strictly specified fates, right? What could be your fates? That's why it's so important. What are other things they are suggesting? This non-gonadal post-embryonic cell lineage of C. elegans are promising for the isolation or characterization of cell lineage mutant. That's a different mutants is coming. When you study genetics, the mutants play a very, very important role. And these lineages are completely known, easy to study, and generate readily recognizable component of the neuronal, muscular, hypodermal, and digestive system. If you have any problem anywhere, you are able to detect that. Okay, next page. Then other thing, since the Horvitz lab is working on this mutant, wonderful mutant, at that time they try to isolate and characterize 24 mutants. Okay, so 24 mutants is a very important role. Among 24 mutants, one of the mutants they are working, next please, it's called lean4. So lean means lineage in the C. elegans, right? Because they determine the different lineage. Okay, so they are isolating, but what does this mutant does? It says the second point, in some of these mutants, cell division fail to occur in the wild type animals. Wild type means there is no mutation, okay? And in other mutants, cell divide that do not normally do so. Maybe it's go and carry on the same thing again and again. That is also not good, okay? So basically they said these mutants differ in specificity of their defect so that it is possible to identify mutation that affect some cell lineage but not in others. You know how important it is. And through this mutation analysis, first they found the lean4, that is the heterochronic gene that previous speaker already mentioned what could be their role. But later study they screened more lines and more. What they found? They found another gene that is lean14. And it has an opposing phenotype of the lean4 mutant that you will learn later. So that is the two things, right? Basically, this novel work about. Go back, next, please. So here, no, okay, okay, fine. I think you already explained. So basically, these are the phenotype or the wild type and the lean4 mutant and this. So what is happen if you are using mutants? So mutants will lead to this. The mutation in heterochronic genes, that is lean4 and the lean14, that cause a temporal transformation of the lineage in many tissue and the cell types. So first, what it does, if a wild type form looks like this, if you make a mutation of the lean4 gene, what will happen? There's loss of function. That is, lean4 is not there. So what will be work? It will elongate it, and all these gonadal eggs come in the center. That is not normal, right? But if you use another mutant that is lean14, what will happen? The adult features will be lost, and that's why I said the precautiously at the larval stage, and resulting is smaller but poorly formed adult. Sorry, it will become adult, not in the larval stage. So later they prove their concept by this experiment, and they suggest the retarded and precocious cell lineage development in lean4 and the lean4 mutants. Why? Because the wild type is going to the both sides, right? You have two color. I think the problem of the base, one is going, this is also not working. Okay, so you can see the two dots, right? One will go to larval 1 stage and go to larval 4 in the later stage. When you make lean4 mutant, is only go on the 1 stage, just making progeny after progeny. But if you go to another mutant, that bypass another thing, that only become an adult. And later, we'll be able to find the lean4 negatively regulates the lean14. That means that much is only known, but how lean4 negatively regulates lean14 is nothing is known. They just so they play the developmental effect and so that's it, and that much information is not there. So go into the next slide. So this is the story of the total, like how from the beginning to the end, now how they get it. Now I'll explain one by one. Now go next, please. So first things they have done, the identification of lean4 as a heterochronic gene. That means play a very important role in the development, and that discovery is done in Horvitz lab. What they shows that in the nematode C. elegans, these heterochronic genes control temporal development pattern in the all the larval stage. In the C. elegans, there is 4 larval stages. It's called L1, L2, L3, and the 4. L1 means first come, L1, right? When you are zygote, maybe you're the baby. Then L4 is the adult, like maybe now we are. So those are the things happen. So one of these genes is LIN4, okay? Discovered by the isolation of the null mutant. Null means you knock it down or you just destroy it. That is called the null mutant. That caused the failure of the temporal development. That means there's no development, right? Some problem is there. And this is the word is supported by these two seminal paper by the Horowitz and the genetics and the cell paper. Okay, go on the next. Now, the second thing what I explained, the LIN4 negatively regulate NIN14. How? This is another seminal paper came by the Ferguson et al. They found that suppressor mutant of the LIN14 genes able to revert. Revert means bring it back to the original place in null LIN4 mutation phenotype. So in fact, they said all the null mutation of LIN14 gene caused an exactly the opposite phenotype of the null LIN4 mutation. That means it's negatively regulating, right, each other. Go to the next. So at that time, the interesting thing came, these two guys, Ambrose and the Rupkin, came and joined the Horvitz lab. That discovery is done, Horvitz lab alone earlier, all this dead gene. And they come and they start working on the cloning of the LIN14 genes. Cloning now is very easy now. Earlier you do on clone, you get on Nature paper, right? It's so difficult because you have to do everything yourself. But when they're working and they publish paper and everything, but they move to their independent lab. When they move their independent lab, the Ambrose focus on the research on the LIN14 and on the other hand, Rupkin study on the LIN14 genes. Okay, that's correct. Now we'll talk about the LIN14 genes by the Rupkin group. By the first author is Rosalind C. By the way, she is the wife of Victor Ambrose, and she is the first author of this. And if you want to know about the authorship dispute and everything, you might maybe read this paper. This is the first time in the happen in the cell, the co-first author is allowed, and they are the make the first time policy. Maybe there will be conflict, maybe husband-wife lab. They send it for the sequencing to confirm it. They do the sequencing. And what they use? They use different 4 closely relative species of C. elegans. They wanted to see among our own thing, this Punjab or not. Those are C. elegans and Bregacy and other, I think, even I do not know in this field. So what they found after that, they have a very much conserved sequence, okay, which is in the dark color, dark board color. And they also found one of the important is the 3' mute here that is complementary to the LIN14 is conserved. That I'll come later, okay? But it's here. So all the this doesn't work. This here you see the red rectangle, all the stories revolve around there. And later they wanted to say, oh, everyone says it might be protein. They look at it has a protein function or not. So what do you do if you want to protein function? If any gene you want to read that codes for any protein or not? How do? You do not know. You look for open reading frame, right? Open reading frame, if you have a sequence, you have a ORF, right? You say where it can read all this transcription machinery. But they do that, neither they didn't find anything. So they say, oh my God, these tiny things do a lot of things, but how it works? This doesn't code for the protein. Go next. So to prove that, they said then it might be the RNA. If it doesn't code for the protein, it can be the RNA. So as you know in the clinical textbook, you always to isolate by RNA with what? With the northern blot, right? So what they isolate? They isolate the RNA. It's basically they cross the ohm, the whole ohm, and they isolate the RNA, right? And from there they use the northern blot with the probe against the LIN14. Okay. What they found the wild type? They found okay, there's a very tiny of RNA is coming, the 22 nucleotide long is here. But when do the rescue experiment, that means in the lean 4 strain, they put the whatever the synthesized the vector, they put it back. So basically, if I take something from your body and if I artificially synthesize and inject you again, you will be expressing the same thing. That is called the rescue experiment. They put it back and they find the lean 4, the smaller fragment even is expressed in much higher order. And more than that, they found another band on the 61 nucleotide long, that is called lean 4L. So they said, oh, it doesn't have a protein, but it might have RNA. But why this so small RNA? Nobody saw on that time. So when you read about the story, they used to do in the tea party, they discuss what is this. Then somebody said, you might look, one of the scientists, the smaller portion of the thing, but Ambrose says, no, this is an artifact. It's it's a junk. It's not true, right? Then they came back and found, oh no, this is indeed a RNA. So what they do once you find the RNA, and the RNA, if you a little bit, RNA cannot work in a linear form. If you want to be RNA functional, it has to be in a secondary structure, or it has to form some kind of loop. So you do not know how our physiological body, blood and everything. If you have RNA, it might be in the coil structure, right, and do all this function. That's why at that time there's no tool how to look at the folding structure. They call it multifold program, and they said maybe the longer form it can have folded structure, but not the linear form. Okay, go next, please. Now I'm asking what the use of this RNA then. Okay, now this discovery is done by 1989 to 1919. That's Ambrose and Rosalind Lee, and that's summarizing what I said. That found 700 base pair fragment of lean 4 gene without the conventional start site and the stop codon. Okay, that's why it doesn't code for the protein. And they introduced the mutation in the putative open reading frame, but the lean 4 function remained unchanged. That means concluding that lean 4 did not encode a protein. In addition, they also found 2 very small lean 4 transcript, that is a 61 and 63 nucleotide length. But Ambrose group is not only working at that time, simultaneously his friend is also working simultaneously, right? Because there are some products. That is the Rapkun. What they are working, they are also looking at the lean 14 function. So now I'll discuss that, then I'll merge later both of them. Go next. So they found that protein, the lean 14, they also wanted to say how this protein is working in the different larval stage, right? L1 or the L4, it's called early and the late. So they have antibody, luckily it's called len 14. Do not worry the myosin, myosin acts as a control. So whatever the cells have, some of the protein we have, no matter what, it will express equally. So this is, we call it control in the experiment, because it has to be respective to something that you have something disease or not. And what they found, lean 4 only expressed in the early stage, not in the late stage. See, there is no band, it's empty on that lane. But they have the other strain, that is lean 4, gain of function strain, as well as the lean 4 strain. And they found it expressed both in the early and the late larval stage. So what does that mean? They are kind of, that doesn't working at the maybe in the protein level, right? So then they are asking, is the lean 4 regulated as a transcriptional or the post-transcriptional level? So what the experiment do? They just do the RNA protection assay. And what they found when they did the RNA protection assay, they found the mainly RNA is present in equal in the both early and late stages. That means it's expressing, right? Protein is not expressing, but RNA is expressing. But what this RNA do, we do not know. So from there suggests this work, there is maybe the post-transcriptional level. Now we have to recall the central dogma. I said DNA to mRNA, but there is another step. It's called the post-transcriptional. Maybe that's why they are doubting that they have a maybe post-transcriptional gene regulation. Go next, please. To do that, they do all this transcriptional assay, right? On the long time when you do the bacterial staining, you have a positive clone or not, what do you have to do? You have to just put the galactoside. If you have a positive clone, I do not know, it might take the green color, right? Because the galactoside is mixed to other things, right? The beta galactoside is coded by the GAL4. So here they move the reporter clone, the reporter line. So far, Horbis Lewin lab also find one of the mutant of the lean 14, and they know the 3' UTR. Okay, sorry, I missed one thing. When I say the transcriptional regulation, the professor already mentioned, the 3' UTR is also very important. It's called untranslated region. They are present in the non-coding portion of the genome. So they also carry some regulatory mechanism. What they do, they took, so in the genetic string, they found this 3' UTR is very important and play very important in the development. So they first put in the wild type strain there in the reporter construct. What they found is expressed both in the L1 but not in L4. Now we remember the Western blot data. When the late and the early and the late. Late there is no protein, right? That's why no signal is there. But they put the same reporter construct of the 3' UTR in the null mutant of the lean 4. Then it will behave crazy, right? And you can see it expresses both L1 and the L14. So now in next, now it suggests I said the lean 4 negatively regulates lean 14. So this is how it's known. Maybe the 3' UTR is negatively controlled by the presence of the lean 4. That much is so far known. So now next please. So now they are puzzled. Okay, they one lab found lean 4. Okay, and it has a developmental defect. Another lab found lean 14. Okay, and it doesn't code for the protein, but you know it has a kind of weird thing. It's work in the post transcriptionally. And but they are in both in Harvard. They are very close, right? And they just make a phone call. That's the real story. And so I have this observation. I have this observation. I say, why don't you come over and send your sequence? And they share the sequence with each other. One share the lean 4 sequence, and the lean 14. And they just overnight they do the alignment, and they found it's the perfect match. Go and it's here when they overlap with the lean 14, that is the bigger gene, right? It has a 3' UTR, and they overlap with the sequence of the lean 4, and they found all the 7 region of the complementary. Then they confirm maybe their mechanism is through by the 3' untranslated region. So that means maybe lean 4 regulating the lean 14 activity by the post transcriptionally by binding with the 3' UTR. So story is not ending here, but so hot. So it's so far they only know it play important role in the larval development. Nothing was known. Now move next. So these are the story. Basically, I say the post transcriptional down regulation by Rofkin group. So in the Rofkin and colleague Bruce Williams, they showed that lean 14 was down regulated at the post transcriptional level, and that in lean 14 3' UTR region was sufficient for the temporal regulation, right? Now this suggests the lean 4 transcript were complementary to the repeat sequence of the 3' untranslated region of the lean 14 genes, right? I'm just summing up. So the two groups share their unpublished data, what I just said. I just reiterate. In June 1992, Ambrose and Rufkin independently came to the same conclusion that lean 4 transcripts were complementary to a repeated sequence in the 3' UTR of the lean 14 genes. Correct? Now go next. So now this is the summary. I think you already explained. I don't want to go back like the mechanism I said. Now next. Now what does it do? Lean 7 you also cover, but I'll cover a little bit more. So far only know this the undefined lean 4 and the lean 14, but they are not conserved. They are only in the C elegans. So they are like aloof for almost 7 years. Then the Rafkun came, that the competition is increasing. They are herding, right? People are interested. They sought RNA. And this paper story is also fascinating, right? The 3 group competing and 3 Nature paper came on same day. Okay. And the Bertel group, their group, and the other group, they are competing with each other. And he is even shocked. Ambrosius wrote, what is this Bertel guy is doing? He even doesn't work in C elegans. And a meeting, he's presenting the data. What the heck he is doing? When the Rediker said there's one paper of the Bertel group also, because they are big shot, right? And the 3 paper came together in the nature of the mechanism. How it works, they discover the late 7, another RNA, and this is conserved from C elegans to higher organism. If it's a conserved, you can see that is very important. We only care for the human, right? We are the human. So that's why there's a conserved. If it's a conserved, does it do anything? So this is the structure of secondary structure, and they found it's combined with C elegans, Drosophila, and the Homo sapiens. And they take our human body different tissue, right? Whatever they want, and do the northern blot with this late 7. And they found the band is everywhere. That's the late 7 RNA on the top panel. But not only one places was there, that is called bone marrow. Bone marrow is our things, everything is happening in your bone marrow. They call that immature B cells or T cells early in the development. So that's why maybe they are claiming this is not present in the lower organism in the right panel. Because when you see the protozoa porifera nidaria, this lean 7 doesn't exist. This only exists in the higher organism. That's why they are concluded maybe that late 7 is a very important microRNA that can control the late temporal transition during the development across the animal phylogeny. This is the groundbreaking story. So one of the very important things came from this scientist called Luisa Cochella. She said the mechanism for controlling protein production was unexpected and entirely new. The quoted is Harvard. Surprisingly, it wasn't really taken super seriously at that time because it was in this womb. Who cares? In the womb, they said, it's the simplest organism. Who cares? People used to talk. And people thought this was something that these funny worms do. They said, oh, you don't do anything. They will behave like this, right? Then the raccoon found the microRNA let7 is present throughout the animal kingdom and include the human. That when the people noticed, right? And started a frenzy that to find all the microRNA that are present in the animals, right? Now this kind of evolution happen, everyone is jumping, right? As the science is like, you want something, then we'll all jump into it, right? Now go to the next. So now this is the summary of this work, how these two microRNA evolved and how their mechanism working. Initially, they found the lean 14 was controlled by the lean 4. And much later, they found, but they play very important in the early developmental stage. Much later, there's a L4 to adult, right? And they found another link called the let7. Let means here lethal, okay? And earlier lean means lineage, okay? Here they inhibit the expression of the lean 14 mRNA, and they say they play very important in the late developmental stage. So two important stories coming. So now I go to next. But the question is, I think he already said, but so what? You find the mechanism, one gene regulating the other gene, either increasing or decreasing. They know what to do with that. But then the bloom came when the first microRNA came in the human. Then other things will come slowly, but you can go next. So what made this guy to give a Nobel Prize? That I am coming. It has to be clinically relevant. That's how it got the Nobel Prize. Okay. Basic science is important, then they translate into the clinic. That's what I am moving now. Why they got the Nobel Prize. But before that, I'll come this discovery. The microRNA will not be famous unless the mechanism is not known. Only mechanism came to know in 1998 when the Craig Mello, the graduate student of Ambrose lab, who got also Nobel, before him, and the Andrew Fire discovered through their studies of the round-arm serial ligands a phenomena called dubbed RNA interference, right? In this phenomena, the double-stranded RNA block messenger RNA so that certain genetic information is not converted during a protein formation. And this silences this gene that is render them inactive. These phenomena play important regulatory role within the genome. How I will explain it later. So basically, the RNA interference is a fundamental mechanism for controlling the flow of genetic information in the cell. Next. So now again, you remember the central dogma. See here, DNA, mRNA, you already know the post-transcriptional mechanism. Now mRNA to protein. If you don't make a protein, you cannot do any function. So if you inhibit there, if you have any bad gene, you cut your umbilical cord. So nothing will happen, right? You're dead. So siRNA can do that. And that is a very important discovery, okay? Now I'll talk about little mode of action, how it is working that he covered. But here the RNA, how the RNAi works. Everyone talk about the RNAi, right? RNAi. So basically these are double stranded and microRNA are single stranded. That I'll also comprehend it later. How they work? It work with the help of the enzyme called dicer, right? And that dicer he talked about it, that concept came only from here, not before, dicer and DGCR. Then basically this dicer cut into the smaller fragment, then bind to the RIX complex, then it makes in the different size of the protein fragment and get degraded, and that's why protein is not anymore functional. So next slide, please. And everything is happening in the cytoplasm. And this is the comparison, but siRNA and the microRNA. So both have the same function. You are just killing the function of your protein, but there is a subtle differences. What are those differences? Because microRNA are single stranded, those are double stranded. Lengthwise, they are much 19 to 25, they are 21 to 22. But what are their action? I'm not reading everything. If you're interested, I can send the slide later that inhibit the translation of mRNA and the cleave the mRNA. Now go next. So now after the discovery, they know the mechanism, how actually it works. From they are stuck on that step, right? mRNA to that is working the post-transcriptionally. But how it is happening with the dicer mediated mechanism or not, they come to later after the discovery of the siRNA. Now go next. So now this is the microRNA biogenesis that Dr. Sandhir already covered, that I'll quickly cover why it is important. Because if you know the biogenesis step, then only you are able to make the drug. How? Because it's a multifactorial procedure. Basically, your primary microRNA is very big. They are transcribed with the RNA polymerase II in your nucleus. and they bind with the microprocessor complex called the DGCR8 and DROSA, and they cleave into pre-microRNA. That's a precursor microRNA. These are 60 to 70 nucleotide long. It is happening in the nucleus. Then you finally it go to the cytoplasm through the exportin pathway. And from there, the additional step is coming. It binds with the dicer1. That is, we learn from the siRNA. Then the microRNA duplex, right, but is converted into the one strand and bind to the argonaute complex and the RIX complex. And one of the strand with complementary, right? They have complementary bind to the 3' UTR of certain protein, and it goes in the two pathway. One is make them a degradation, and another just knock it down. So this is how it working. Now go next. So now we also covered a little of the oncogene. So now it's okay, microRNA is fine, the mechanism not, does it have any relevance and any disease or not? So it's a cancer, right? So microRNA can function as a tumor suppressor or the oncogene. Tumor suppressor means it suppresses the tumor formation. Oncogene, it have much oncogenic properties. That's why it's saying, or called the oncomete, that microRNA is overexpressed in the cancer. So if you found these two mechanisms, that microRNA either functions as a tumor suppressor or the oncogene, in both of the cases, you have aberrant expression. It makes more tumor, but how? They do not know, because what is happening in the cells, it doesn't know. So from this slide, you can get an idea that microRNA mutation or the misexpression correlate with various human cancer and indicates that microRNA can function as tumor suppressor or the oncogenes, and microRNA have been shown to repress the expression of important cancer-related genes and might prove useful in the diagnosis treatment of the cancer. Now it's coming much more important. Next, please. And this is he already covered. That this is the treatment of microRNA and what kind of disease it can be regulated is covered. Next, please. And if you want to develop the microRNA therapies, these are the very normal thing. You do the discovery-based research, and then you see, there are all these clinical parameters is fine or not. And then you finally try the large animal. Then you finally go to the preclinical trial. In the US so far, the very previous data in 2020, that means patent has been filed in the microRNA research. Now this list is even much more. Go next, please. Now, forget about the left side, is much more complicated. Just focus on this monkey side. So this is very important breakthrough. And that time, one of the microRNA called microRNA 122. And this microRNA 122 play a very important role. If you have a higher cholesterol, they have shown this microRNA, if you inhibit by the called locked nucleic acid mediated mechanism, it's called anyway to degrade your microRNA, that can decrease your cholesterol level. That is first time shows that the green yellow monkey, that's called the primate, right? MicroRNA is there. So when you use that LNA modified microRNA against microRNA 122, inject into the monkey, and you see your cholesterol level is decreasing. So this is one of the proof of the preclinical studies, right? And the later on of the studies because of microRNA 122, that can also silence your chronic hepatitis C virus. That also got Nobel, by the way. Chronic, the people who discovered the hepatitis C virus, very dangerous virus. The very replicative. You just put that is the replication, means it makes the another copy, means another copy of us. And that's why we get infected and we die, right? The loading is increases. where the will be same. You're just making them full. You put that in a patient sample, and you can see the microRNA overexpression is going up. That means you can relieve from the lung cancer. Okay, next please. That suggests that microRNA can be have a potential target. And these are the companies throughout the world who are working on the microRNA and the RNA therapeutics, right? And one of the things I saw earlier that the microRNA wanted to, this is the companies from Denmark, Santaris Pharma. They already developed it and they reached in the defined even the clinical 4 level. Next. And I think this is the last slide. I think this is after the Nobel is declared. How happy are they, including their dog? So basically, Ambrose said after the Nobel Prize, what the microRNA really end up revealing for us is a way that parts our genome can communicate with other parts of the genome, right? One part of the genome to the other part of the genome. So coding versus the non-coding, right? Clear? And this is the significance of this discovery of the microRNA is that it allowed us to aware of very complex and nuanced layer of regulation whereby gene in our cells talk to each other. Isn't it clear? It is nuance, right? That means what is the function, what is the known, and they eventually find the proper mechanism and what could be the transcriptional applicability, and it can go to clinic or not. And that I think the Nobel Committee is forced to give them a Nobel. They talk about they will get a Nobel maybe 10 years back. They didn't get it, but eventually this year is giving. So thanks to them and their research and the basic research. With that, I'll congratulate them for the Nobel and thank you for listening to me. And okay, you have a question?
Thank you very much. We are now ready for the question. So both the speakers will be
They understand everything.
One or two questions can be.
Nothing is silly.
and there were other people working in the same area as you said, for lab papers. How come these two were picked up for the Nobel?
Because they are the only one who continue with that research. Because the Ambrose was continuing with that lean 4 that is later turned out to be microRNA, and the Rupkin work on the lean 14. I think always there is in the lab a lot of postdocs or researchers go. They don't care later. They do their postdoc, they go for a job, but they continued.
Nobody else started their work.
At that time, it was not very fancy. Yeah, and that time they
lucky to get them connected also.
And at that time, they even do not know to work on this tiny thing. Luckily, they are in the US. That's why a lot of people did not get a tenure, because they are not that productive. If you don't work in the exciting thing.
He said something very interesting.
Say?
He was not given permanency in his position because he could not probably get a grant also.
So the tenure means, let's say you have assistant professor position here, and you are in a temporary, right? 5 years. NCBS has it, IISc has it, and some of the IISER has it. You are not productive enough. Let's say you are not teaching good. You did not bring the grant. You might be sacked in your job. That happened to them. That also happened with the mRNA vaccine.
In 41?
Yes. So lean 41 is one of the gene that is controlled by the LET7.
And it comes later in the development stage.
That Kevin, the larval 4 stage to the mature one, and that the LET7 play very important role.
That's what is conservative also, conserved in all species.
What normally when we study developmental biology, throughout we are studying we are studying its expression. Most of them is like that only. But what is the basic role of conservative genes? Like you were saying कि middle में वो नहीं है, then it was getting conserved in humans, lower पे conserved है, but in the initial some of the models.
It's called the seed region in the microRNA. So they are 21 to 22 nucleotide long. It's big because why you need that? You kind of, let's say if you're cold, right? We like to hang around together, right? Sitting. So that is the principal work on the transcription factor. You know what I think he mentioned the transcription factor discovery, and it's the end of the world, right? But if you find the binding of one transcription factor, multiple transcription factor bind together and act together like attacking together. So link RNA has 21 to 22, but not everything is required. They call the seed sequence. 5 to 7 nucleotide is more than enough. That 5 to 7 nucleotide can be used for targeting the drug. That's why it is so specific.
And perfect matching is not required also
Yeah.
for miRNA. That's what they also want to say.
So
that's why they are able to bind to multiple partners.
So that is another drawback. So one microRNA can control thousands of genes also. That is the specificity of the issue. That's why maybe they are facing a problem, but not for siRNA. siRNA is most specific.
Making it even more deeper.
But it's good. I think come on.
Although there were features, but we managed to get the Zoom working, and thanks to Dr. Ravi also.
Oh. One. Yeah.
We have a couple of things which is not cool.
Hello, may you ask a question?
Please wait, sir, please.
Hello. Hello. Dr. Singh, कैसे हो? How are you?
Yes, fine.
I will be sending you the pictures tonight because I was busy with the lecture. that's why.
Okay.
That's why I sent you a short message in the morning.
Yeah, it's good. Thank you.
So how's everything with you? Back in Germany?
Yes.
Okay, great.
Nice to hear it from you. Thanks.
Thank you. Then see you soon. Then. Dr.
Okay.
Kia is here. Dr. Kia.
Hello. Thank you, Dr. Rajendra Singh, for being with us. It was
Thanks
wonderful
also
that
from
you
my side.
attended the whole of it.
Handy. Yes, I did.
Thank you.
आपको परमानेंट वेन्यू दिलवाना है मैंने डाल के दिए ठीक है
आराम से करना धीरे दिखाना खींचना मत उसको खींचना नहीं उसको आना है तो काट दिया
यार